far, we have discussed the basic processes taking place during photosynthesis. It includes light reactions and the dark reactions. Photosynthesis is a process which is very sensitive to any kind of biotic or abiotic stress. Any change in the environmental condition affects the efficiency of this process. At the same time, efficiency of photosynthesis is directly correlated with the crop yield. Overall plant growth, development and yield are directly proportional to its efficiency to carry out photosynthesis. In this context, it becomes important to know the factors which influence the rate of photosynthesis and thus indirectly regulate plant yield. The environmental factors affecting the process of photosynthesis follow the law of limiting factors as hypothesized by the British plant physiologist F. Blackman in 1905. According to this hypothesis, when a process is influenced by a number of factors, the rate of the process is limited by the slowest factor. For example, photosynthesis can be influenced by both carbon dioxide concentration and light intensity. By increasing the CO2 concentration at a fixed intensity of light, the rate of photosynthesis can be increased up to a particular concentration of CO2, after which no further increase can be noticed. At this point, by increasing the light intensity to another fixed level, the rate of photosynthesis can be increased with increase in CO2 concentration. In this example, the intensity of light is the limiting factor for the rate of photosynthesis. The implication of this hypothesis is that at any given time, photosynthesis can be limited either by the light intensity or by CO2 concentration, but not by both the factors. The main factors affecting the rate of photosynthesis are light intensity, CO2 concentration and temperature. Now, let us see how each of these factors affect the rate of photosynthesis. Light. Light is essential for photosynthesis. Photosynthesis does not take place in dark. The sun is the main source of light energy. Both intensity and quality of light are important for photosynthesis. Light intensity. The intensity of light influences the rate of photosynthesis. When other factors are not limiting, the rate of photosynthesis increases almost linearly with increase in light intensity. At low light intensities, as light intensity increases, the rate of light dependent reactions and therefore photosynthesis generally increases proportionately. A clear straight line relationship is observed. More is the number of photons of light falling on a leaf, the greater is the number of chlorophyll molecules that are excited and more ATP and NADPH are generated. As light intensity is increased further, however, the rate of photosynthesis is eventually limited by some other factors. So, the rate plateaus, meaning that there is no further change. At very high light intensity, chlorophyll may be damaged and the rate drops steeply. At extremely high light intensity, when leaves are unable to utilize the absorbed light, the rate of photosynthesis declines by a phenomenon called photoinhibition. Photoinhibition is caused by the absorption of too much light, which inactivates and damages the reaction center of photosystem II. In the dark, there is no photosynthetic CO2 fixation taking place in the leaves. Rather, CO2 is given off by the plant because of respiration. By convention, photosynthetic carbon assimilation is considered as negative in this situation. As the light intensity increases, photosynthetic CO2 fixation increases until it equals CO2 released by respiration. The light intensity at which the rate of photosynthetic carbon dioxide uptake equals the rate of respiratory carbon dioxide released is known as the light compensation point. Plants do not survive below the light compensation point. Light quality. Different wavelengths of light, that is, different qualities of light, are not equally effective in photosynthesis. Different plants show different rates of photosynthesis under different quality of light. In green plants, only red and blue light are effective for photosynthesis. Green light is reflected or transmitted. Therefore, it does not play any role in photosynthesis. Light of wavelengths longer than 700 nanometers is not effective for photosynthesis for green plants. Maximum photosynthesis occurs in the red and blue part of the spectrum. For example, photosystem 1 absorbs energy most efficiently at 700 nanometers and photosystem 2 at 680 nanometers. Light with a higher proportion of energy concentrated in these wavelengths will produce a higher rate of photosynthesis. Carbon dioxide. The atmosphere is the chief source of carbon dioxide. It contains only 0.03% of the gas by volume. It is a very small amount. Therefore, carbon dioxide remains a limiting factor. Carbon dioxide is the key inorganic molecule required for photosynthesis. As it is normally present in the atmosphere at very low concentrations, increasing carbon dioxide concentration causes a rapid rise in the rate of photosynthesis, which eventually plateaus when the maximum rate of fixation is reached. Then other factors may become limiting. The increase is more rapid up to 1% of carbon dioxide concentration, but it slows down beyond this point. Higher concentrations have an inhibitory effect on photosynthesis. Temperature and the rate of photosynthesis. A suitable temperature is necessary for photosynthesis. There are three cardinals of temperature for photosynthesis. First is minimum temperature. It is minimum temperature at which the photosynthesis starts. It varies a lot between different photosynthetic organisms. For example, minimum temperature for many lichens is 20 degrees centigrade, while it is minus 35 degrees centigrade for some conifers. Photosynthesis hardly starts at about 5 degrees centigrade in tropical plants. Desert plants like cactus can carry out photosynthesis even at 55 degrees centigrade. Second is optimum temperature. The optimum temperature for the process of photosynthesis also varies greatly. Photosynthesis increases with rise in temperature up to 25 degrees centigrade. This increase follows Van 't Hoff's law. According to this law, the rate of chemical reaction doubles for every rise in 10 degrees centigrade. This is true only if light or carbon dioxide is not the limiting factors. The third is maximum temperature. It is the highest temperature at which photosynthesis can take place. There is an initial increase in the rate of photosynthesis at this temperature, but this is soon followed by a decline. Higher the temperature, more rapid is the decline. The decline may be due to inactivation of enzymes, destruction of chlorophyll molecules, and damage to other proteins. Although the light dependent reactions of photosynthesis are not affected by change in temperature, the light independent reactions of photosynthesis are dependent on temperature as they are mostly catalyzed by enzymes. As the enzymes approach their optimum temperature, the overall rate increases. It approximately doubles for every 10 degree centigrade rise in temperature. Above the optimum temperature, the rate begins to decrease and finally stops as the enzymes get denatured. Water. Water is one of the raw materials used in photosynthesis. The amount of water utilized in photosynthetic reactions is quite small. Less than 1% water is absorbed by the plant. Therefore, it cannot be a limiting factor directly, but the water content of the leaf often acts as a limiting factor indirectly. It maintains the turgor of the assimilatory cells. The rate of photosynthesis decreases in the cells which have lost their turgor. The loss of turgor of guard cells closes the stomata, thereby reducing the rate of photosynthesis. Thus, inhibition of photosynthesis is observed at different degrees of water stress. Leaf anatomy. Many factors of leaf anatomy affect the rate of photosynthesis. These factors influences the diffusion of carbon dioxide through the stomata. They also have effect on the amount of light reaching the chlorenchyma cells of the leaf. These factors are thickness of the cuticle and epidermis and size, position and distribution of the stomata. Chlorophyll content. Chlorophyll is essential for photosynthesis. Etiolated plants and non-green tissues do not show photosynthesis. The green cells produce starch in variegated leaves. As we know that the process of photorespiration competes with the process of photosynthesis. It is because of both carboxylation and oxygenase activity of the enzyme rubisco. If rubisco combines with carbon dioxide, it results in photosynthesis. However, if rubisco combines with oxygen, it results in photorespiration. All those factors which can increase photorespiration will result in decreased photosynthesis as well. So, it is necessary to know about the factors which increase photorespiration and thus inhibit photosynthesis. Photorespiration rates are increased by altered substrate availability, that is either lowered carbon dioxide or increased oxygen. Factors which influence this include the atmospheric abundance of the two gases, the supply of the gases to the site of fixation, that is in land plants whether the stomata are open or closed, and the length of the liquid phase, that is how far these gases have to diffuse through water in order to reach the reaction site. For example, when the stomata are closed to prevent water loss during drought, this limits the carbon dioxide supply while oxygen production within the leaf will continue. In algae and plants which photosynthesize underwater, gases have to diffuse significant distances through water, which results in a decrease in the availability of carbon dioxide relative to oxygen. It has been predicted that the increase in ambient carbon dioxide concentrations predicted over the next 100 years may reduce the rate of photorespiration in most plants by around 50%. Another factor which influences photorespiration is increased temperature. Increasing temperatures reduce the solubility of carbon dioxide in water more, thus reducing the concentration of carbon dioxide relative to oxygen in the chloroplast. For example, at 15 degrees centigrade, carbon dioxide to oxygen ratio is 0.046, while it becomes 0.037 at 35 degrees centigrade. It has been found that this situation favors oxygenation over carboxylation activity of rubisco. What are the different methods of measuring the rate of photosynthesis? There are a few key methods to calculate the rate of photosynthesis. These include measuring the uptake of carbon dioxide, measuring the production of oxygen, measuring the production of carbohydrates, measuring the increase in dry mass. IRGA. Uptake of carbon dioxide can be measured with IRGA, that is infrared gas analyzer, which can compare the carbon dioxide concentration in gas passing through a chamber surrounding a leaf or a plant and the carbon dioxide leaving the chamber. Measuring photosynthesis via the production of oxygen. Quantification of oxygen released in the presence and absence of light by a plant is a good approach to measure photosynthesis and respiration. The amount of oxygen evolved can give idea of the efficiency with which photosynthetic process is going on. This can be measured by using an oxygen electrode. The oxygen electrode works based on the principle of Clark, that is, using a polarographic measurement of the electricity that flows between an anode and a cathode, and it is sensitive enough to detect oxygen concentration in the order of 0.01 millimole. A thin Teflon or polythene membrane which is permeable to oxygen and a piece of paper beneath the membrane are usually placed over the electrode surface in order to protect them and to provide a uniform layer of electrolyte between the anode and the cathode. Chloroplast suspension is kept in the sample chamber and is shown light. As a result, oxygen is evolved and is quantified by using a computer software. Measuring photosynthesis via the production of carbohydrates. This is a crude method where a disc is cut out of one side of a leaf using a cork borer against a rubber bung and weighed after drying. Some days or even weeks later, a disc is cut out of the other half of the leaf, dried and weighed. Increase in mass of the disc is an indication of the extra mass that has been stored in the leaf. This is very simple to do and enables to investigate plants growing in the wild. However, there are several inaccuracies in this method. Measuring photosynthesis via the increase in dry mass. Dry mass is often monitored by the technique of serial harvest, where several plants are harvested and dried to constant weight and weighed. This is repeated over the duration of the experiment. It can give a measure of the surplus photosynthesis over and above the respiration that has taken place. Investigating the light dependent reactions in photosynthesis. In 1937, Robert Hill showed that a partial reaction of the electron transport chain using DCP-IP dye could be used to investigate the rate of oxygen evolution and thus the rate of photosynthesis in thylakoids of isolated chloroplast. The reaction is now known as the Hill reaction and is used still today to determine photosynthetic rates in chloroplast preparations. The rate of decolorization of the dye DCPIP in the Hill reaction is a measure of the rate of light requiring stages of photosynthesis. Isolated thylakoid membranes are prepared for this experiment. The dye reagent used in this experiment is DCPIP, that is 2,6-dichlorophenol indophenol. It is blue when oxidized and colorless when reduced. DCPIP accepts electrons between the electron chain components plastoquinone and cytochrome. The electrons are ultimately derived from water. Chlorophyll fluorescence. Measurement of chlorophyll fluorescence is proving to be a wonderful tool to study plant physiology and plant stress. Chlorophyll fluorescence techniques appear to be a very powerful tool for the non-destructive study of photochemical and non-photochemical processes within thylakoid membranes, chloroplast, plant tissues and whole plants. Chlorophyll fluorescence is the light re-emitted by the chlorophyll molecules while returning from excited to non-excited or ground states. Light that is absorbed by a leaf follows three competitive pathways. It may be used in photochemistry to produce ATP and NADPH, or it can be re-emitted as fluorescence or dissipated as heat. These processes compete with each other to use the absorbed energy. Chlorophyll fluorescence can measure most types of plant stress. Chlorophyll fluorescence can be used as a proxy of plant stress because environmental stresses like extremes of temperature, light and water availability can reduce the ability of a plant to metabolize normally. This means that less energy will be utilized for photosynthesis and more energy will be emitted as fluorescence. This relationship is used as an indicator of photosynthetic energy conversion in higher plants, algae and bacteria. The Kotsky effect. Upon illumination of a dark adapted leaf, there is a rapid rise in fluorescence from photosystem II, followed by a slow decline. Since it was first observed by Kotsky and co-workers in 1960, this is called the Kotsky effect. The increase in fluorescence is due to photosystem II reaction centers being in a closed state. Reaction centers are closed when they are unable to accept further electrons. This occurs when electron acceptors downstream of photosystem II have not yet passed their electrons to a subsequent electron carrier, so they are unable to accept another electron. Closed reaction centers reduce the overall photochemical efficiency and so increases the level of fluorescence. Transferring a leaf from dark into light increases the proportion of closed photosystem II reaction centers. So, fluorescence levels increase for 1 to 2 seconds. Subsequently, fluorescence decreases over a few minutes. This is due to a number of reasons like more photochemical quenching, in which electrons are transported away from photosystem II due to enzymes involved in carbon dioxide fixation, and maybe because of more non-photochemical quenching, in which more energy is converted to heat. FV by FM ratio. Fluorimeters are used to measure chlorophyll fluorescence in the field. FV by FM ratio is the most commonly used chlorophyll fluorescence parameters in the world. The FV by FM test is designed to allow the maximum amount of the light energy to take the fluorescence pathway. It compares the dark adapted leaf in pre-photosynthetic fluorescent state called minimum fluorescence or FO to maximum fluorescence called FM. In maximum fluorescence, the maximum number of reaction centers have been reduced or closed by a saturating light source. In general, the greater the plant stress, the fewer open reaction centers are available and the FV by FM ratio is lowered. FV by FM ratio is a measuring protocol that works for many types of plant stress. It is a measurement ratio that represents the maximum potential quantum efficiency of photosystem 2 if all capable reaction centers were open. An FV by FM value in the range of 0.79 to 0.84 is the approximate optimal value for many plant species, with lower values indicating plant stress. FV by FM is a fast test that usually takes a few seconds. Chlorophyll content meters. Chlorophyll content in intact leaves can be measured by using a chlorophyll meter. These are instruments that use light transmission through a leaf at two wavelengths to determine the greenness and thickness of leaves. Transmission in the infrared range provides a measurement related to leaf thickness, and a wavelength in the red light range is used to determine greenness. These instruments and scales have been shown to correlate to chlorophyll chemical test for chlorophyll content, except at very high levels. Chlorophyll content can also be measured in isolated chloroplast by dissolving in acetone or methanol and measuring absorbance at different wavelengths. Increase in the chlorophyll A by B ratio is an indicator of stress condition in plants.